Welcome to The Explainer. So today we're taking a really fascinating journey straight into the microscopic world to decode the absolute bedrock of genetics, Mendel's Laws of Inheritance. Now, you might be wondering, why does this matter right now? Well, in our modern era, understanding the biological code of life is literally more relevant than it has ever been. So we're going to neatly unpack these tiny, almost kawaii-level cellular rules that perfectly govern exactly how physical traits are passed down from one generation to the next. Here is our incredibly simple roadmap for today. First, the law of segregation. Second, visualizing alleles in meiosis. Third, independent assortment. And fourth, monohybrid versus dihybrid crosses. Honestly, you only need to master two main concepts today before we synthesize them completely into the larger picture. Section 1. The law of segregation. Okay, let's just dive right into this by starting with the single most fundamental rule of inheritance.
This is Mendel's first law, better known as the law of segregation. Officially, it states that the two alleles for a heritable character segregate during gamete formation, and they end up at different gametes. But let's put that simply. If an organism has two alleles for a single trait, they absolutely do not stay bundled together when it's time to reproduce. Actually, they separate completely.
Section 2. Visualizing alleles in meiosis. Now, reading a biological rule is one thing, right? But let's see exactly how this microscopic split physically happens inside a cell. Let's look at this really beautiful process. Here we have a separation of alleles involving a homozygous dominant genotype. Basically, that means we have two capital Ps representing a purebred purple flower. First, you'll see the genetic material undergo replication. Then, as we move down through the stages of meiosis I and meiosis II, you can literally watch those replicated chromosomes perfectly split apart into separate, single allele gametes. It's so neat! Each resulting cell gets exactly one capital P. Now, what's really fascinating here is what happens when the individual isn't purebred. Look at this heterozygous genotype. This is an F1 generation purple flower, and it's carrying one dominant capital P and one recessive lowercase p. The crazy thing is, even when we mix a dominant and a recessive allele, the cellular machinery still segregates them flawlessly into a 50-50 split. Half the resulting gametes get the capital P, and half get the lowercase p. No exceptions. The rule is totally unbreakable.
So, the crucial takeaway so far is that a single trait separates cleanly. But come on, that begs a massive question. What happens when we observe two traits at once? Do they stick together as a package deal, or do they act alone? Let's challenge our brains a bit and bridge our first concept into the next beautiful layer of complexity.
Section 3. Independent assortment. And that brings us perfectly to Mendel's second law, which handles multiple characteristics happening at the exact same time. Mendel's second law is the law of independent assortment. It states that each pair of alleles segregates independently of any other pair of alleles during gamete formation. In plain terms, a seed's color and a seed's shape don't boss each other around. The alleles that decide if a seed is yellow or green sort themselves out entirely on their own, completely separate from the alleles that are deciding if that seed is going to be round or wrinkled. And this brilliantly illustrates exactly how chromosomes can arrange themselves in two different, really cute combinations to make that happen. Notice the possibilities during meiosis I here. On the left, dominant alleles might line up together. But over on the right, they might just mix and match. Because they sort independently, these possibilities lead to four uniquely different gamete types: capital Y capital R, capital Y lowercase r, lowercase y capital R, and lowercase y lowercase r. Section 4. Monohybrid versus dihybrid crosses. All right, let's see how all of this builds into the actual traits we observe by putting everything we've learned head-to-head for a grand summary. Let's walk through the strict parameters on both sides of this. On the left, we've got a monohybrid cross. This observes just one character, determined by one gene, something like flower color, and it involves crossing two true breeding individuals with different phenotypes. Simple enough, right? But on the right, we have a dihybrid cross. This observes two characters at once, like seed color and seed shape, crossing double true breeding individuals. For example, crossing a completely yellow round seed with a completely green wrinkled seed. Now just let your eyes guide you from left to right here. On the left, the monohybrid cross gives us a very simple four-box Punnett square. We're just tracking one single trait, like tall versus dwarf plants. But on the right, look at that glorious multicolored 16-box matrix for the dihybrid cross. Because of independent assortment, the gametes mix to create major visual variety. You don't just get the parent combinations, you get entirely new combinations, like yellow wrinkled seeds and green round seeds. It's like a giant puzzle coming together.
Which brings us to a final provocative thought. How does this microscopic, cellular dance dictate the infinite variety of life we see around us every single day? It is truly awe-inspiring to realize that everything in our modern era, from cutting-edge CRISPR gene editing all the way to personalized medicine, is built entirely on these adorable but very strict biological rules. Thank you so much for diving into the fascinating world of genetics with me on this explainer. Keep wondering and keep learning.